right. Our next presenter is Dr. Vasu Sharma. He's an assistant professor at the University of Minnesota. Her research interests are in the areas of irrigation and water management, water and nitrogen fertilizer interactions, crop evapotranspiration, soil water dynamics, variable rate irrigation, and cover crop water management. Vasu earned a master's and PhD degrees from the University of Nebraska-Lincoln. So with that, Vasu.
All right. Thanks, Fabian, for the introduction. My name is Vasudha Sharma. I'm the irrigation specialist at the University of Minnesota in Soil, Water, and Climate Department. And I know this is nitrogen management conference, and same as Chris's, I will be talking more about water than nitrogen, but a little bit about nitrogen as well. So the topic is variable rate irrigation and nitrogen management for corn production, and I'll be talking about some data results that we got from last 4 years from this ongoing project. So there's one more year this year that is left for that study, but I'll be showing you some prelim data from that project. But before we start with research, how many of you deal with irrigation here in the room? Great, we have good. For those who don't, some background information about irrigation, because irrigation is not that we do in whole state. We have very concentrated area where we do irrigation. And this is the map showing you in 2018 from DNR active agricultural irrigation permits. And most of these permits are concentrated in the central sands region of the state, which is mostly glacial outwash sands in the central Minnesota, and around 600,000 acres of crop irrigation, which is close to between 4 to 5% of total cropland in the state. So not big number in terms of the percentage as compared to total croplands, but amount of water that we are using in this area is huge. So the major crops that are grown are corn, 48%, some soybeans, dry beans, potatoes, canning crops, and others. So we'll be concentrating on corn for our research because that's highly produced crop in this area. And most of the water that we use for irrigating these crops comes from groundwater. So 93% of the irrigation water applied to these acres comes from our groundwater resources and some from surface water. In the recent years, we have seen a drastic increase. So from USDA data from 2002 to 2017, we have seen approximately 34% increase in these irrigated acres. And the main reason, as you know, is the climate change. We are seeing dry summers mostly from last 3 years as we experienced. So more irrigation is coming into practice, in particular in these areas. And one reason we only irrigate these areas is because of coarse texture soils. We are irrigating the sandy soils that do not hold much water for a longer period of time. So for profitable crop production, we need to irrigate these soils. And the problems associated with it. We know irrigation is important. Crop production is not feasible or profitable without irrigation. But at the same time, there are issues that are related to not just irrigated acres, but overall everywhere. But if we look at this data, this is the map from on the right from Department of Health, looking at the drinking water wells, which have nitrate levels above 10 parts per million or milligrams per liter. And you can see most of these are in the sandy soils region of the state, which coincidentally are the irrigated regions as well. So that's where we need to look at how we efficiently manage our irrigation, because we need to produce the crop, but at the same time, we have to deal with environmental impacts of it as well. And just not the water quality, which has been an issue from very long time, but also water quantity, as we are seeing climate change in the recent years. And some press, these are some articles that were published last years focusing on irrigation and how, because of the dry years, some of the areas in that central region have been over pumping water over the permit. So water quality is a big issue, but now we are seeing, as we are moving into this climate change scenarios, we are looking at more of water quantity issues as well, which we have witnessed in Western states. I'm from Nebraska, my PhD is from Nebraska, and we have those issues back there and in most of the Western states. And Minnesota was different because we have lots of water, but things are changing and we now have to think about other than water quality, water quantity as well. So about the project that I'm going to discuss with you today, we are focusing on two major corn production inputs, which is water and nitrogen, because we know that they are interdependent. We have good data from many years of research about the nitrogen response, but we don't have data for water response. So we know what is recommended nitrogen amount for irrigated corn in sandy soils, but we do not have the data to tell what is that right amount of water that your crop needs in order to produce profitable crop, but at the same time, without any environmental impact. So that's the study. This study comes in where we are looking at water, but not just water, looking at nitrogen as well, because they are interdependent. If we are putting more water, we are leaching the nitrogen. If we are putting less water, nitrogen uptake will be low. So we are trying to gather or collect the data to make some recommendations over time, same as the nitrogen recommendations. Just to give you a background, like this is study focusing on water and creating water response curves at different nitrogen rates. So the main objectives are first to evaluate how irrigation and nitrogen interact with each other. Is there any interaction effect? And how they are affecting yield, crop evapotranspiration. Crop evapotranspiration is how much your crop is using the water that you are applying as irrigation or what is coming into the rainfall. Crop water productivity and efficiencies, both water and nitrogen. And then based on these data sets, we develop best management practices for water management in the central Minnesota. And then third component of the study is to look at how we can use some sensing data, both proximal sensor and remote sensing, to develop some in-season corn, both water and nitrogen diagnosis methods. And I'll be showing you some of the data about that too at the end. So this is the 5-year study. We started in 2020. 2023 was the 4th year, and this 2024 growing season is going to be the concluding year of this study. We have 2 sites in central Minnesota. One is at Westport, which is in northeast of Pope County, and Becker in Sherburne County. We have a sampling research farm up there. Both sites, we selected these 2 sites. One thing is they are both in central sands region, so both have sandy soils, coarse textured soils, irrigated sites. But at the same time, they are in vulnerable water quality zone as well, as described by MDA. The experimental design. So for the first 3 years, 2020, 21, and 22, we had 4 irrigation treatments, full irrigation, and I'll explain that to you in the next slide what full irrigation means. Then 75% of full irrigation, 50% of full irrigation and rain-fed. But in 2023 and this last year, we are slightly changing these irrigation treatments, and our irrigation treatments for last 2 years will be 125%, because we wanted to see if we over apply water than what our soil can hold, what would happen. And then 65%, 100%, 65% and rain-fed irrigation. However, for this year, for the last year, 2023, when we needed most water from mid-June to mid-July, at both of our sites, our irrigator was not working. So we ended up with 100% with the same treatments last year as well, because we tried to apply more water, but we ended up with broken pivot at both locations at the same time. So we had that, and very dry time, we were just dependent on rain rather than irrigation for most part of those mid of June to mid of July. But the nitrogen treatment stays same. So 6 rates, 0 to 350 pounds of nitrogen and 70 pounds nitrogen increment. So total 24 combination treatments and replicated 4 times. So each site have 96 plots. And we are using split plot design. So this red box is a replication, and within each replication, we have 4 irrigations. Within each irrigation, we have 6 nitrogen rates. So the 100% irrigation, coming back to that, what we are doing, so some of you might ask, like, how would you know 100% irrigation is 100% irrigation if you're using irrigation scheduling tools such as checkbook method? So checkbook method might be, the ET numbers that the checkbook method are using is wrong. So the 100% is actually not 100%, it is 125%, but we are not relying on any irrigation scheduling method. We are relying on our soil moisture sensor data. So each of the plot have soil moisture sensor, and we know what is the field capacity or water holding capacity of our soil. So we are monitoring that soil moisture continuously, and whenever in our 100% plot, we are close to our irrigation trigger, we irrigate it back to field capacity. So that's our 100% irrigation. And once we know 100% irrigation, we reduce it to 75%, 50%, and no irrigation for other treatments. If we are applying water above this field capacity line, that is more drainage or runoff. If we are going below this irrigation trigger, which is for corn, usually 50% of plant available water, we are stressing the crop. So in terms of measurement, we are measuring water below the root zone using suction cup lysimeter. So we have in each of our plot at both sites, lysimeter installed permanently at the location. So there's a lot less disturbance in this data as well, because the lysimeters are installed permanently. And I'm not going into detail of that, but if you have any questions, I can answer those after the presentation, like how this instrument works. And then we are collecting soil moisture also from 3 replications in this study using neutron probe. Neutron probe is a soil moisture sensor, which is for research, it is a standardized method of measuring volumetric water content that uses radioactive material and throws out neutrons in the soil and measures magically the volumetric water content. I can explain that if anybody have questions, how does this work? And then plant sampling and soil sampling throughout the growing season. We have 3 plant sampling campaigns to understand nitrogen uptake under these different irrigation rates and nitrogen rates. And at the end of the season, we also collect soil sample for whatever is left in the soil, the nitrogen. And then for the remote sensing and proximal sensing part, we use different kinds of sensors to measure all the data we can. We use leaf clip sensor, crop circle phenome, UAV, remote sensing to collect thermal images, multispectral images in order to develop some diagnosis method, which are in season for both nitrogen and water. So starting with results, so irrigation, this is the Westport site on the left, right side is the Becker, the 4 years irrigation. The highest irrigation was in 2021. And this is mostly, as you know, we are measuring soil moisture. So mostly dependent on the rainfall. When we get the rainfall, we do not irrigate. Our soil moisture is enough. But when we do not get the rainfall, we do irrigate. So these numbers are based on our soil moisture sensor measurements. But they do not follow exactly the rainfall patterns all the time. So now you see if these are the 4 years of rainfall at both sites, and on the top of each graph, you see the long-term average. In 2020, which was the wetter year, the growing season, so this is from planting to harvesting. At Westport, we got around 18 inches, at Becker around 16 inches. However, in 2021, we got more water at Westport, which was a dry year. So the total water could be more, but if you look at just this part, which is mid-June to kind of beginning of August, that's the critical part for water. So in this time frame, we did not get much water in 2021. So you see the orange line at both locations was at the bottom. So that's why in 2021, we had maximum irrigation, because before starting of June or after August, we do not irrigate in Minnesota, mostly because we get rains. But that's the most important part, when we do not get rain. And that's what happened in 2021. And in 2020, if you look at that, in both locations, we got good rains in that part. Corn yield at Becker. So lots of data, I know, but I try to do the averages at the bottom. So this is all the data for 4 years. So different bars are different colors are different years. And we see we got very good response with nitrogen in all years. So in irrigation 1, 2, 3, and 4, all 6 rates of nitrogen. And as we increase the nitrogen, our crop yield increases, and then it flattens out here. So significantly looking at the statistical data, we did not find any significant difference in crop yield at higher 3 nitrogen rates in terms of yield. And if you look at the irrigation data, this is the average data of 4 years, same response to nitrogen. And we have an irrigation response as well. As we decrease the irrigation, our yield decreases, but statistically, we did not see any significant difference in grain yield, particularly at higher nitrogen rates for 175% of irrigation. In terms of evapotranspiration, so evapotranspiration is crop water use. So we calculated that using water balance approach. We had all the components of water input and water outputs, and we calculated ET as residual. And the same results for evapotranspiration on as we increase the nitrogen, we do not see that big increase in ET at higher nitrogen rates. We see that all 3 higher nitrogen rates have almost same amount of ET, but if we look at the water, which is this is the average of 4 years, as we decrease the irrigation, the evapotranspiration also decreases, but no difference in ET between 100 and 75%. That means even if we were applying less water in 75% irrigation, the evapotranspiration was similar to 100% irrigation. And that reflects the corn yield as well. So we did not see, because ET was same, there was not big difference in yield as well between 75 and 100% irrigation. And this is yield at Westport. Same response same as Becker. We did not see a significant difference between 100 and 75. But at Becker, since we got more rain as compared to Becker, we also did not see on 4-year average difference between 75 and 50%. So 50% was also very comparable to 75% at Westport site. So there are some site differences as well between in terms of yield and ET. Interestingly, at Westport, we did see a significant difference in evapotranspiration between 100 and 75%. At 75%, our evapotranspiration was slightly lower, significantly lower than our 100% irrigation, but no difference in 75 and 50% irrigation. All right, now the good results, which I was waiting for. So that's all the data, or I just throw the data at you just to show how much data we are collecting and what kind of data. But this is the yield, this is what we are focusing on to develop the relationships, like how much water we put in the system and how much grain yield we are going to get. The 6 lines here are 6 nitrogen rates. And as you can see, all of these rates, as you increase the water, so this is total water, both what comes in rain and what we are putting in irrigation. As you increase your water input, at each nitrogen rate, your corn yield is going to increase, but at the top, it's going to flatten for all of these irrigation and comes down at higher water input, which is very interesting. So we see same as nitrogen response curve, we see this water response curve. This is 4 years of data. And the maximum yield at Becker site is approximately 226 bushels per acre. And what does this tell us? That high availability of water or nitrogen, if you look at the higher end rates, the top lines, the decline is more prominent than at the lower. It's more flat, but it is coming down. So which means that if you are putting more water or more nitrogen into the system, there is more vegetative growth, but less grain yield. So more water or more nitrogen is also not good as lower amounts. And then evapotranspiration as a function of total water. So how much irrigation or precipitation that you put into the system reflects to how much your crop is using. So you increase your water input. That doesn't mean that your crop is going to use all of that water, right? So we see this. So this is a diagonal line, which is 1-to-1 ratio, which we think like if we increase the water, our data will be all on top of this line, which doesn't happen in actual world. So we have, there is a point where after that, this green line is going to go away from this diagonal line, which shows that there are inefficiencies in the system. So as you put more water, that all of that water is not going to be used by the crop. And from more data, this is the relative yield versus crop evapotranspiration. What we found that And this is very important number, and I'm very excited with the 4 years of data, I get to this. This is what I wanted, is equating an ET value of 18.5 inches, which comes from this data, which reflects the maximum yield. So you need 18.5 inches of evapotranspiration to produce maximum crop yield in this region. And when you put that into this equation, that comes out to be 23.6 inches of water input, which is combined precipitation and irrigation to produce maximum crop from 4 years of data. So the idea is to put more and more data as nitrogen response curves. They are based on 30 years of past data, right? So you put more and more data into these graphs, you get your R square is going to increase and you get more accurate numbers. But this is from 4 years of study. And this is true for all nitrogen rates. These are yield evapotranspiration functions. So as you increase the seasonal ET, which is increased by seasonal total water input, your crop yield increases, but at the top you see at higher nitrogen rates, you see that decline as well. So not always increasing ET will reflect into increasing yield. And this is evident from our nitrogen uptake data as well that we collect throughout the season. So this is grain plus stored nitrogen at R6 crop growth stage. And I had to separate the years, and I don't have 2023 data yet, but if you combine the years, it doesn't make sense. So I separated the years because each year was different because of the rainfall and the weather. We see that for at all irrigation levels, as you increase, as the seasonal evapotranspiration increase, the nitrogen uptake increase, but every time at higher ET, there's a drop in nitrogen uptake. So ET, the crop water use doesn't always reflect crop nitrogen uptake. There is that point, the maximum point after which the nitrogen uptake declines. And that's true for if you have more irrigation, more ET, your N uptake is not the maximum. The maximum N uptake comes before that maximum water. Then we cannot forget about nitrate leaching, because we talked about all the production and profitability, but we can't miss the environmental impact of that. So we collected, as you know, all the lysimeter data. This is the 4 year of data. And you see the high standard deviation bars and very high numbers in 2020. And that was the first year of our data collection. And at that time, we installed the lysimeter. So there was a lot more disturbance in the system. So there's a lot of variability. And that's one reason we installed permanent lysimeter, so that we have less disturbance over the years. And that's what we see in 21, 22, and in 23. Our standard deviation bars are low. And with the dry years, all these 3 years were really dry in the summer. So we did not see much nitrate leaching as well, not above 10 parts per million. And on average, this is average of 4 years. What we see is as you increase the nitrogen at each irrigation rate, your nitrate leaching increase. So as you put more nitrogen into the system, your nitrate leaching is going to increase. And that's not just because of water irrigation input. It also is a component of how much nitrogen is available there to be leached down. So lower nitrogen rates have lower leaching, higher nitrogen rates have higher leaching. And if you move from I1 to I2 to I3, you see there's a slight decrease. If you just compare the last 3 bars in each of these, N4, N5, and N6, as you decrease the irrigation, on average, the nitrate leaching also decreases. So to go into more depth of it, I looked at each year separately. I forgot to put the years on top. This is the wettest year, 2020, then 21, 22, and 23. On the x-axis is the total water that we were putting in the system, both irrigation and precipitation. On y-axis is the nitrate leaching. In the first year, didn't make any sense because there was so much irrigation. And irrigation was, at Westport, we applied only 2 irrigations, total 3.2 inches. So not much to tell for 2020, but in last 3 years, I am seeing a trend, and I put the equations to show you the R squares, which are really great. And we see that it starts high. As you increase your total water input, it decreases, but then at very high water input, it increases again. So this matches with our yield, nitrate uptake, everything. At high water, high ET, the system is not performing very well or not profitable. And same for the environmental impact. We see that increase in nitrate leaching at high water input, but there's that sweet spot right here, which is where we have least nitrate leaching as well. So the key findings are both yield and ET are dependent on irrigation water applied and nitrogen applied. And what I'm looking, which is not here, is the site is totally different. I did not show you Westport data. It doesn't follow the same pattern. It is slightly different what we see at Becker, and maybe next year I will show you that data as well. But there is a site difference because of the difference in weather. So how the precipitation falls, what timing, how frequent, it all matters. How the leaching would happen, how the nitrogen uptake would happen, and what the crop yield would be, and the total amounts of water and ET as well. Few more slides. As I promised, I'm going to show you the remote sensing data, what progress we are making. Very prelim data, just 2023 season. So we collected thermal and multispectral images to understand how we can use those data sets for in-season water and nitrogen management. And focusing on just the water, we have the nitrogen data as well, but for this presentation, I'm just going to show you some maps of what we are looking at in terms of water stress detection. So we had thermal data, both canopy, above canopy and below, and air temperature data, and we calculated crop water stress index. This is the same study I showed you, and you see one crop water stress index is very high water stress. That means low water input, and more blue is low crop water stress index. Your crop is less stressed. And we did see a good correlation of crop water stress with our irrigation treatments. You see the 0% are more on the higher side of stress, whereas the irrigated plots are on the lower side of the index. So there's a promising result, but statistically, we did not see any significance of irrigation or nitrogen based on crop water stress index. So we were curious, so we added NDRE, which is normalized difference red edge, and this is our function, and then we see a significant difference of irrigation when we use this index rather than just crop water stress index. And this is the function of crop water stress and NDRE, and here we see a significant correlation of this function with our irrigation treatment. So there is some promise of collecting those thermal images and multispectral images and how we combine these data sets for in-season diagnosis. This particular image is average of many dates throughout the season. And then if you are interested more in irrigation, looking at results, what we are doing, this is one project where we have several projects focusing on irrigation. We have several extension and outreach efforts that we do. Minnesota Irrigators Program, and I'll be showing you in the next slide details about that. But YABA is here, NRCS, RCPP, which MDA is leading, and University of Minnesota, along with 20 soil water conservation districts. We are partnering on this effort where there's cost share money for growers to apply for more water conservation practices. We are done with our first round and hopefully get more funding from NRCS in the second round. And then in irrigation and nutrient management field days that we do throughout the season in the summer. So Minnesota Irrigators Program, this is a 3-day program focusing on basic to advanced irrigation knowledge for our growers in the state. This year, we are going to do this, and this is the 3rd year of this program in March 16th, 13th, and 20th in Central Lakes College in Staples. So if you're interested, you can register for that. We have limited seats. We have only 25, and I think we are close to 15 registrants right now, so which is good. So if we have 10 seats open, if you're interested, you can register for this. And if you do that, there's an incentive that if you are Minnesota water quality program certified, you can get an irrigation endorsement through this program, which will help you in applying for more funding for water conservation implementation. And that would be all. I would like to thank everyone who helped with this project. The funding agencies are co-PIs, Fabian Nushin, our grad students who are working very hard. So most of the graphs and data analysis that you see are done by the grad students, thanks to them. And then field crew, we have some field crew members here. Thanks to you for helping us with all the field work, and then other collaborators on the project. Thanks.
Right, thanks, Vasu. Let's give her a hand. And we have time for questions, and I see a number of questions here. So we'll start with the first one. Is there any long-term data suggesting that irrigation is depleting groundwater supplies?
That's a great question, and I'm also very curious about knowing that. I don't have that data, but I think DNR has observation wells throughout the state, and I don't know how accessible that data is, but if we have access to that data, we can do some modeling and look at the groundwater levels in the state, but I'm not focusing on that right now. But I'm sure there is data to look at that.
And I suspect that the combination of 3 dry growing seasons with more need for irrigation probably kind of drove some of those concerns. Yeah. All right. So was plant available nitrogen contributions from irrigation water accounted for in the amount of nitrogen applied to the crop?
No, that is not accounted for, but after we started this study, from last 2 years or 3 years, we are collecting the water samples from the irrigator and looking at how much nitrate is in there, but we are not accounting for that in our recommendations in this study.
How does an irrigating grower without continuous soil moisture sensors maintain a soil moisture level at 75% of field capacity?
That's really a good question. So for that, we have different tools that you can use. We are focusing on developing a web application, which is ongoing, and we are trying to input more data into it, called IMA, Irrigation Management Assistant Tool. So if you have some basic information about the soils in your field, that what kind of soil, how much water that soil holds, maybe some initial testing is required, and the soil don't change like in 5 years or 10 years or even more. So the holding capacity calculation has to be done just once. So if you have that information and you use the tool, it gives you pretty good recommendation about irrigation. 100% irrigation, and then you can reduce that to 75%. So soil moisture sensors is a plus. So this is to check if the model that you are using is working well. But if you do not have soil moisture sensor, that irrigation assistant tool works very well. And I have comparison data as well from our research studies to look at that.
Very good. Do you anticipate that irrigated acres will dramatically increase due to climate change?
So the USDA data that I showed you, we see an increase, and with the same climate, if we see more dry years like we have seen in last 3 years, I can say we would see an increase in irrigated acres in the state.
Could you briefly elaborate on vegetative growth becoming excessive to the detriment of grain yield? How would silage figure into that?
That's a good question. We have not looked at silage corn, but yeah, so what we have seen when I went to the field in August, we have seen big plants, like more than 10 feet tall, in our 350 pounds of nitrogen and high irrigation rates. But that did not reflect in our grain yield. So that's where we, like, more water or more nitrogen input would make crop look, more vegetative growth, but less grain yield. But that may be applicable to silage corn, where you need more vegetative growth, right? But I do not have any data to look into that yet.
About devices used in that trial?
So we use soil moisture sensors, the neutron probe, which is, I don't know if you want more details, but I can help you with that after this. But neutron probe for soil moisture sensor and suction cup lysimeters for nitrate leaching.
Very good. What were the average nitrate leaching levels at the nitrogen fertilizer rate considered to be optimum, meaning not excessive?
That's a great question. I have the data, but I do not know who would make the recommendation of what that threshold would be, like after which it is. For Department of Health, 10 parts per million is the limit, but in terms of pounds per acre, I do not know. What would that be?
Yeah, I mean, so we do have the data in the work that you're doing there. It's just a matter of kind of calculating where that economic optimum
Yes.
end rate is and where because you have for each nitrogen rate, you also have the amount of loading that or
Yes.
loss that happens.
Yes, we have all the data to calculate what that optimum N leaching would be, where we have maximum economic output. And yeah, so we can calculate that, but I have not done that yet.
So it's an ongoing project. That's
Yes.
actually one of the data points that I'm really interested in looking at.
Yeah.
All right, well, very good. Let's give Vasu a hand.